So what we have done in hematology on blood physiology is that compositions and functions of blood, about plasma proteins and erythropoiesis. These topics have already been done. Composition and functions of blood, those who are joining newly, for them I am just revising that all these things you have a very basic idea you should be having from your school level. Composition and functions of different components of blood. So what is blood? A fluid connective tissue. Okay, you all know this. Blood is a fluid connective tissue. It is a connective tissue. Connective tissue means it is mesodermal in origin. So these things I think need not be repeated here. Any connective tissue is mesodermal in origin. Blood is a fluid connective tissue flowing through the blood vessels. The human vascular system is a closed vascular system, and blood is contained in the blood vessels. That is what are the names of the blood vessels? Arteries, veins and capillaries. The thinnest one are the capillaries. About the vascular system, it is being discussed in the cardiovascular classes on cardiovascular system. About the vascular system, we are discussing here on blood. So blood is a fluid connective tissue. It is just more viscous than plain water and all. It is mostly liquid. Obviously, what are the components of blood? Plasma, fluid part is plasma, and defibrinogenated plasma is called something called the serum, ACRUM. Defibrinogenated plasma is called the serum. Very grossly speaking, it's plasma minus plasma proteins. Plasma minus plasma proteins is serum, and blood contains non-fluid elements. Those are the cellular elements of blood, more specifically they are called formed elements. Why it is better to call formed elements rather than cells of blood? Because at least one component is there which is not a complete cell. What is that? That is the platelets. Platelets are formed from something called megakaryocytes. They are precursors, and platelets are actually cell fragments, not a whole cell. So instead of cellular elements of blood, it is better to call formed elements of blood. The term formed elements of blood are used. Among the cellular components of blood, red blood cells, white blood cells. White blood cells are of again two types based on the basis of presence of or absence of granules, granulocytes and agranulocytes. Granulocytes are namely, they end with the suffix "phil", neutrophil, eosinophil, basophil, and agranulocytes are lymphocytes and monocytes. And okay, we'll come to that another thing later. So those are the types of blood cells. RBCs, they have no as such subtypes. Platelets also don't have. Among the formed elements of blood, we classify only white blood cells as granulocytes and agranulocytes. And composition of plasma, we have mostly it is water, more than 90%, and rest of it, except this 92%, rest of it is mostly plasma proteins, most of which is again something called albumin. The most abundant plasma protein is albumin, others are globulin, fibrinogen. There are a lot of plasma proteins, over maybe more than 200 have been identified till now, but the most abundant plasma protein is albumin. Okay. So and also it contains obviously some microorganisms will be there always. It is a human body, so some circulating nucleic acids, which nucleic acids are there in the nucleus of the cells, but some circulating nucleic acids, very traces of them are found in the blood as well. And blood's functions are, as you all know, it is mainly transport of oxygen and nutrients, but it also acts as for transport of hormones. And one main function which we will read in a separate section called immunity, immune defense functions, mainly carried out by the WBCs. Okay, immune defense functions. And blood components help to maintain the acid base balance of the body. pH buffering function of blood is another function, but main function which is without blood, nutrients and oxygen vital to the tissue, vital for living. That in the human body, carriage of nutrients and oxygen is via blood, okay, and waste removal obviously through the kidneys. About the plasma proteins, the total concentration concentration 6 to 8 gram per dl in blood. Whenever we speak of different components of blood or different these numerical figures will come. This is the amount of this blood component, this is the amount of this is this figure, number of RBCs, number of WBCs, amount of hemoglobin. You will find all throughout there are some definite units are used, like total number of RBCs. Theoretically, what is the number of RBCs? We know around 5 millions. It is in males 4.5 to 5.5 millions, but what is important is 4.5 to 5.5 millions in how much of blood? Something microliter or cubic millimeter. Same thing, 1 microliter or cubic millimeter. So blood cell counts are in microliter, or same thing is cubic millimeter. Microliter is in the SI system, cubic millimeter is in the CGS system. So blood cell counts we express in microliter or cubic millimeter. Now can we express it in deciliter, milliliter? Of course, but the figures will become not very comfortable figures to make the figures look, because if we try to express it in milliliter, then we have to multiply it by 1000. So that the figures look good. So for that reason, the per microliter of blood is chosen for the counting of blood cells. What is the amount of WBCs? Likewise, it is 4000 to 11,000 again per microliter. Amount of platelets, it is 1.5 lakhs to 4 lakhs per microliter. But hemoglobin, amount of hemoglobin, it is expressed in deciliter, gram per deciliter, 12 to say so there is it per dl or 100 ml, and wherever you will find 100 ml, you will instead of that you may find the term percentage, like hemoglobin, same thing, this is same thing. So these, why I am just discussing it like this, you will get whenever you will find in books, you go through textbooks, you go through problems, you face the exams, you have to express yourselves. You just have to know the standard units in which what figure is chosen, written in what units. So for hemoglobin we use per milliliter, per dl, or that is same thing is per 100 ml, or it is just percentage. 12 to 15 gram percent means gram per 100 ml. For cell counts we use, blood cell counts we use per microliter or cubic millimeter of blood, either per microliter or per cubic millimeter. So likewise, plasma proteins are in gram per dl, and normal plasma proteins are 6 to 8 gram per dl. Now if you argue can it be expressed in milliliter, of course it can be expressed, but it will become 0.06, 6 will become 0.06. To express it in to make the figures look good, we express it like this, 6 to 8 gram per dl. Obviously it will be 0.06 if it is ml. If it is in microliter, the figures will still become less, it will be so many zeros after the decimals. So to avoid those, it is in dl, per deciliter of blood 6 to 8 gram, and most of it is albumin, okay. Almost out of 6 to 8, almost 3.5 to 5 gram per dl is albumin, and plasma proteins are formed in, where they are formed? Obviously in the embryo, connective tissue is mesodermal in origin, blood components are mesenchymal origin in embryonic level at the mesenchyme cells, and then the liver takes up. Mostly they are synthesized from the liver, the reticuloendothelial cells from the liver. Only one type of plasma protein, gamma globulin, it is synthesized from lymphocytes, B lymphocytes, which are again a type of white blood cells. After the white blood cells are there, from there the gamma globulins are formed. So albumin, globulin, fibrinogen, among globulins, these are separated by electrophoresis. We have classification is like that. On the basis of electrophoresis, there are alpha globulins, beta globulins, gamma globulins. These gamma globulins are formed from B lymphocytes. alpha and beta globulin from, except the liver, some may be from spleen and bone marrow also, but mostly most plasma proteins are from the liver. Functions of plasma proteins: coagulation. See, here also they are telling these are the same, the percentage of albumin is greatest, percentage of globulins are lesser than that of albumin, but fibrinogen, prothrombin and other plasma proteins are only traces. Functions of plasma proteins: this is about hypoproteinemia. So hypoproteinemia, just again I have told this, but those who are new, whenever you find this term, you know this is related to blood. Whenever you find this suffix, this is related to blood. Proteinemia means it is speaking of the concentration of proteins in blood. Hemoglobinemia means it is speaking about the concentration of hemoglobin in blood, amount of hemoglobin in blood. Proteinemia, if it is hyperproteinemia, less proteins in blood. Hyperproteinemia, it is about more proteins in blood. There are various causes, you don't need to worry about all the causes right now, but there are various causes due to which there may be hyperproteinemia, there may be hyperproteinemia, like when a lot of blood is lost in hemorrhage. Okay, hemorrhage means loss of excessive amounts of blood. If blood is lost, obviously plasma proteins will also decrease due to malnutrition, due to dehydration and all. Due to dehydration, what may happen? Due to dehydration, that means water is coming down, water content is coming down, so relatively as if the protein concentration is going up. That is a cause of relative hyperproteinemia. So you don't have to worry about causes. As you go on learning in medical science, you will get various causes of various things, but just know for now as beginners, this EMIA term, whenever you will get, it is speaking about the concentration of the different components of blood, and there are various causes of hypo and hyperproteinemia. Plasma proteins, obviously they have different functions. Some plasma proteins like fibrinogen, albumin is the most abundant plasma proteins, and it has transport functions. Thyroid hormones in the body, like one example is thyroid hormones in the body are carried by albumin, they are bound to albumin and they are carried in the blood. So role in transport, again plasma proteins, some role in defense mechanisms, some role in coagulation, fibrinogen and prothrombin, they have some role in the buffering action, that is regulation of acid base balance and maintenance of osmotic pressure. Again, those who have not attended general physiology classes, transport mechanisms or diffusion, osmosis, these things are being discussed. Osmosis is diffusion across what we know from school days, semi-permeable membrane. That means movement of solvent in the direction of where there is less concentration of solute. Now to prevent this movement, this movement can be prevented by applying a pressure on the side where the solutes are more in the direction in which the solvent is moving. If pressure is applied, then that pressure, that pressure which will just counteract that movement is called osmotic pressure. Now plasma proteins, these are various physiological, that means important physiological significance of the body, the role of maintaining that osmotic pressure to prevent movements always. If always we don't want the movements, this fluid movement in towards where in the human body is the semi-permeable membrane, the cell membrane is the semi-permeable membrane. So this osmotic pressure which will help or which will see to it that the fluid is not always moving inside and outside the cell whenever not required, that is maintained, that osmotic pressure maintained. The major contribution to that is by the plasma protein. So these are the various plasma proteins and various functions. There are other plasma proteins also, fibrinogen, leave it, acute phase protein, these terms you will get acquainted. Again, when in different discussions, acute phase protein means whenever there seems some acute disease condition. So these are all these, not only fibrinogen, it is also only one of the coagulation factors also, but it is an acute phase protein. Is induced release of acute, these terms you will get, interleukins and all, these we'll again discuss when we discuss immunity, interleukins, cytokines and all. Okay. So different plasma proteins except these transferrin plasma cellular plasmins are names of other plasma proteins, transport of iron, transport of copper. So these are names of different plasma proteins. Transferrin, ceruloplasmin, albumin is a major plasma protein, but so many 200 to 300 plasma proteins have been identified. You don't need to know the names of all. We will get through these names as we go on, but see, free hemoglobin is transported by something called haptoglobin. So this is another example. So these are the various types of plasma proteins. Osmotic regulation, as we told, osmotic regulation, plasma proteins have a role in osmotic regulation. These are amphoteric molecules, that means they help in pH balance, buffer balance. They have carboxy terminals and amino terminals, and both in acidic pH and alkaline medium, they have to maintain the pH of the, because pH has to be maintained within a very narrow range. To maintain homeostasis, maintain balance, physiological balance, pH has to be maintained in a very narrow range, and plasma proteins have contribution to that. Hemoglobin has also contributes, but plasma proteins have contribution to maintaining that acid base balance in the body. Leave all these again classification of plasma proteins, not very extensive but quite large list of some plasma proteins functions and all. You don't need the large list, but just that some different plasma proteins are helping in they have different functions throughout in various systems in the body, okay, like haptoglobin, again binding of hemoglobin and carriage of hemoglobin. Ceruloplasmin, transport of copper, we saw in the previous slide also. C-reactive protein, C-reactive protein is something which is increased in infections. It is called C-reactive protein, or in short CRP. So don't need the extensive list right now, just there are so many plasma proteins, their concentrations differ, but the most, if we should not call it the most important one, but the most abundant one is the albumin, okay. So and they can be clinically, they are helping in different clinical scenarios also, because we have to know when hypoalbuminemia occurs, hyperalbuminemia occurs, when albumin is increasing, to know about the affinity of drugs having same affinity for albumin. Some drugs are having same affinity for albumin if they are administered together. So that knowledge will help us as medical professionals. If they are administered together, then if already one drug is more bound to albumin, another drug is required to act, then if already one drug is given, another drug administering another drug, but that function will not be served because it will not get enough albumin to bind with. So these things, knowledge about plasma proteins have various clinical applications. Okay, now we had also discussed about erythropoiesis, that is formation of RBCs, and the formation of blood cells or formation of most cells are originally from what cells? These stem cells, which are the first precursors, the preliminaries of most types of cells. So stem cells can differentiate into themselves, can differentiate into other cell types, and they act as precursors. So be it erythropoiesis or leukopoiesis, be it RBC production or WBC production, the origin is from the stem cells, and the site is at least in adults, the site is the bone marrow. We will come across these different terms: totipotent, unipotent, multipotent. Unipotent is still not not used in such all standard textbooks, but totipotent, pluripotent, these are standard accepted terms. Totipotent is at the zygote level ability to differentiate, at the zygote level, after the formation of zygote, ability to stem cells which are able to differentiate into different types of cells at the zygote level, and these are among these potent stem cells, these are the most potent. In that way we can call it, these are the most potent. And pluripotent is, then it can give rise to the germ layers. Pluripotent cells can give rise to the different germ layers. If you are discussing, I don't know how much of embryology is being discussed these days in anatomy, but these things are more discussed in the embryology classes. Multipotent ability to differentiate into different cell types, but not as that means as functional or as functionally diversified as totipotent. but they still have the differentiate into different types of cells. So these are just these terms you will come across as you read these topics. Whenever you have to describe about erythropoiesis, not that you have to write these things or tell these things, but these terms, to understand the topic you have to preliminary idea about these types of these terms, otherwise it may be difficult to understand. So again, this blood something, there are different theories of these formations of blood components and blood cells, but something called the hemangioblast. Blast cells are, again, blast cells are precursor cells. Blast cells are nothing but precursor cells to mature cells. So there is something called the hemangioblast. They are saying one theory is saying from the hemangioblast at the mesodermal level, from the hemangioblast there are cells differentiated. Some are differentiated to form the endothelial lineage. We will form, endothelium means the inner side of the inner wall of the blood vessels, and some types of cells will, that lineage will develop for the development of the blast cells. That is the, either some cells will go for endothelial specification and some will go for hematopoietic specification, or the, those are called the hematopoietic stem cells. Hematopoietic stem cells, but the origin is from the hemangioblast. Again, another theory is telling from the whatever is the preliminary hemangioblast, that is all are forming the endothelial cells, and from the endothelium these hematopoietic stem cells are arising. So these are different theories, worry much about that. You just, I am discussing a bit to just to make you know that ultimately whatever, wherever from the blood cells are arising from, the precursors must be hematopoietic stem cells. Okay. Now there can be, some are long term hematopoietic stem cells, some are short term hematopoietic stem cells, but ultimately they are from the hematopoietic stem cells, whether they are originating from the endothelium or they are separate from the endothelium in origin, direct from the hemangioblast, directly from the hemangioblast, is still a matter of debate. Different theories are going on about it. So from the, what happens from the stem cells, from the long term cells, from the short term cells, various precursors are named as blast forming units, colony forming units. That means ability to give rise to a burst of different types of cells, or ability to give rise to a colonies of certain types of cells. These terms will come, burst forming units, colony forming units, with different prefixes and suffixes. Like you get a term CFUE, that is colony forming unit for erythrocytes. So you don't, again you don't worry about many terms at this, that means all these terms at this level, you just will just simplify it. Like for erythropoiesis, see so many types of, so many lineages. For erythropoiesis it will go to colony forming unit. It is the same for megakaryocyte and erythrocytes. So those precursors for platelets and erythrocytes together written as CFU-MEG. That means these colony forming unit will again divide into different lineages. For megakaryocyte, it may be for megakaryocyte, it may be for erythrocyte. So from the CFU megakaryocyte erythrocyte, it is to BFU, burst forming unit of erythrocyte, and ultimately the erythrocytes will develop. For the platelet lineage, it will develop from platelets, and all these what is CMP? CMP is common myeloid progenitor. Myeloid means most cells which are associated with these RBCs, and most cells with innate immunity. Innate means from birth. Myeloid lineage. Lymphoid lineage for the lymphocytes. Lymphocytes are mostly associated with this adaptive immunity. We will discuss this about immunity. We will have separate sections, as in your MBBS curriculum now immunity is given as a separate section in the hematology syllabus, that means in the physiology syllabus as a separate section. You will find it in the competencies in your physiology syllabus. Those who are new, I will tell for them just to search out CBME 2024 on the internet, CBME 2024. Just go and search it, you will get your detailed syllabus for all the subjects which you will have to face in the MBBS curriculum. For now, you concentrate on, for the first phase, anatomy, physiology, biochemistry. So I missed this is about just where from the where erythropoiesis is taking place. Again, in the first 6 weeks of life, that is after the zygote stage, as the embryo is developing, it is from the yolk sac. In the embryonic stage, from the yolk sac, okay, and then liver and spleen takes it up from about 6 weeks of stage, about 6 weeks to 6 months in the liver and spleen, and then in the bone marrow. But in the adults, in the children and the adults, it tends to precede the liver and spleen, but the mostly it is the bone marrow. And in adults after 18 to 20 years of age, say, in the long bones, it in the bone marrow. In case of the long bones, it occurs only at the ends of the bone marrow, but also other areas like vertebrae, ribs, sternum, pelvic bones, but not the whole long bone, at the ends of the long bones after 18-20 years of age. So, this is a gradual, that means transformation from where, not transformation, that means a gradual shift in the area from which this erythropoiesis will occur. There is nothing much to write. These slides are all about from either they are from some standards textbooks or from the internet. So, almost all information which are shared here, you will get whatever textbook you read on physiology, you will get that. Obviously, try to follow a textbook which is meant for MBBS physiology curriculum, because other courses have the textbooks are written according to the syllabus for those courses. So, follow any physiology textbook which is written for the MBBS curriculum. So, this is about the areas of erythropoiesis. Erythropoiesis have different stages. We had already discussed pro-erythroblast. Blast cells again are precursor cells. Erythroblast, the precursors for erythrocytes. Pro-erythroblast, then the different stages: basophilic, polychromatic and orthochromatic erythroblast. Then comes the reticulocyte and the erythrocytes. These polychromatic, orthochromatic, basophilic erythroblast, orthochromatic erythroblast, polychromatic, basophilic, orthochromatic. Yeah, the books you read, you will may find the terms early normoblast, late normoblast, early intermediate and late normoblast, same as polychromatic, basophilic and orthochromatic erythroblast respectively. Erythroblast and normoblast are actually practically identical things. Erythroblast precursor for normal erythroblast, they are also calling them normoblast. So, early, intermediate and late, but what you have to note is that which stage RBC, mature RBC is a non-nucleated stage, and after the orthochromatic erythroblast stage, the nucleus is exuded. Reticulocytes, just the immediate precursors for erythrocytes, are also non-nucleated. Up to the stage of orthochromatic erythroblast, these cells are nucleated. Now, these are days of MCQs. So, when is the up to which stage, at which stage the nucleus is lost? It is lost after the stage when the formation from orthochromatic erythroblast to transformation to reticulocyte. At that stage, the nucleus is exocyzed or it is exuded from the cells. So, nucleus, and hemoglobin starts appearing again in the intermediate normoblast, which is similar to which is the same as this basophilic erythroblast stage. At that stage, hemoglobin starts appearing, and the whole process takes about, say, 7 to 10 days. Some books are writing 1 week, 7 days. Some books are writing up to reticulocyte stage, it is 7 days, but then it takes 1 to 2 days more from reticulocyte to the mature erythrocyte. So, we may safely write that it is 7 to 10 days the whole process of erythrocytes. Gradually nucleus size is decreasing, cytoplasm is increasing, nucleus size is decreasing, and ultimately it is exuded at the stage of during the transformation of orthochromatic erythroblast to forming the reticulocyte. Hemoglobin starts appearing at the intermediate normoblast stage, which is the same as basophilic erythroblast stage. Sorry, and the size of the mature erythrocyte is around 7.5 micrometer is the diameter. RBCs are shaped, shape of RBCs are biconcave shape, the shape is like this. So see, the cell size is gradually decreasing, the nucleus is gradually decreasing, the cytoplasm gradually actually increasing in amount, and hemoglobin starts appearing at the stage of intermediate normoblast or basophilic erythroblast. So what is the functions of RBCs and all? RBC carries, so this hemoglobin which actually is the for the binding of oxygen is mostly carried by hemoglobin. Erythropoiesis is influenced by different factors. Main factor for erythropoiesis, the factor which mostly induces erythropoiesis, main factor inducing erythropoiesis is hypoxia. Hypoxia means lack of oxygen at the tissue level. There is a hypoxia related factor or the hypoxia inducible factor. That factor is responsible for stimulating erythropoiesis by secretion of a factor called erythropoietin from where? From the kidney. Erythropoietin is a hormone which is secreted from the kidney. It comes to the bone marrow and then acts so that erythropoiesis can occur. So erythropoiesis is there are other factors, but low oxygen levels in the body are the primary factors for it. And what other factors influence? There are dietary factors like vitamin B12, folic acid, these are needed for erythropoiesis, but the primary factor which is inducing, the primary factor is whenever oxygen levels are low, erythropoiesis is increased to a great extent. There is a hypoxia inducible factor has been identified for. So other factors, some like these things you will come to know. Some beta adrenergic stimulants, adenosine, one of the factors, some cobalt salts, these also induce erythropoiesis to some extent. Alkalosis means decrease of acids or increase of bases in blood, chemical bases in blood, some hormones, thyroxine, but hypoxia is the main factor. And what is the thing? Some decreased secretion of erythropoietin is due to some adenosine, like adenosine facilitates erythropoietin secretion, but adenosine antagonists like theophylline, it is or hormones like estrogen, these are decreasing the secretion of erythropoietin. But mostly this single factor hypoxia and induced hypoxia induced increase of erythropoietin is the most important factor. And dietary factors, whatever we receive from diet, like if there is vitamin B12 deficiency, folic acid deficiency, then RBC formation is hampered. Okay. So androgens, these are about the hormones. Dietary factors, iron, copper, different vitamins, mostly vitamin B12, some hematopoietic growth factors are there. So these are all about the factors influencing erythropoiesis. There is a factor called intrinsic factor of Cassell's intrinsic factor, secreted from the parietal cells of the stomach. Probably you are already studied about it in gastrointestinal physiology. So help in the absorption of vitamin B12. Vitamin B12 is needed for erythropoiesis. Intrinsic factor of Cassell helps in the absorption of vitamin B12. In that way, it is helping in erythropoiesis as well. So about leukopoiesis, so again this is the same thing which we, this is an extensive one. This is that how from the short term, long term and short term hematopoietic stem cells, they are differentiating into different lineages. This is how the megakaryocytes, the erythrocytes, the leukocytes, and from the common lymphoid progenitor only the lymphocytes. So origin of similar origin like erythropoiesis, leukopoiesis also is involving obviously the stem cells. So one lineage, myeloid lineage, is going to megakaryocyte erythrocytes and the granulocytes among the leukocytes, and from the common lymphoid progenitor it is the lymphocytes. So this is about the just similar to erythropoiesis, leukopoiesis, but the granulocytes are from the myeloid lineage. Just this difference you have to remember, and the lymphocytes are from the lymphoid lineage. Granulocytes and monocytes formed in the bone marrow. Obviously, most are in the bone marrow. Only some lymphocytes are from the, they are originating from the bone marrow, but their development is in the other lymphoid tissues, like there is a section of the lymphocytes called the T lymphocytes which develop in the thymus. Thymus is one type of lymphoid tissue. So, there are various lymphoid tissues. Some plasma cells are, what are plasma cells? Plasma cells, this is not to be confused with blood plasma. Plasma cells are one type of cells which are immunity formed from the associated with immunity formed from the B lymphocyte cells. So, those are called the plasma cells. So, lymphocytes and plasma cells, these are not originating, all are originating in bone marrow actually, but development takes place, they migrate to the lymphoid, other various lymphoid tissues like thymus. T cells are developed in the thymus. So, white blood cells, again these are divided into granulocytes and agranulocytes, neutrophils, eosinophils, basophils, lymphocyte and monocyte. Neutrophil main function is combating acute bacterial infections by a process called phagocytosis, which you should be knowing about. Phagocytosis, they engulf these invading agents and form the phagolysosomes and then help the body to get rid of them. So, in acute infections, acute bacterial infections, neutrophils numbers are increased. Neutrophils numbers in peripheral blood are increased. They migrate from the peripheral bone marrow to the peripheral circulation. So, the function of neutrophils is to combat in acute bacterial infections. Neutrophils are also called polymorphonuclear leukocytes. Although neutrophils, eosinophils, basophils should together be called, but the term, this PMN term, polymorphonuclear nucleoside, is mainly restricted to the neutrophils. It is usually restricted to the neutrophils, but ideally speaking it should cover neutrophil, eosinophils and basophil as well, polymorphonuclear, because the nucleus is of formation. Those who have attended, those who have joined late, we will try to revise these classes at some time, but those who have already seen the cells under the microscope can identify or can appreciate better. Why polymorphonuclear neutrophils? Nuclei are 2 to 7 lobed. So, this variation in shape, so they are called polymorphonuclear leukocytes. It is difficult to see. Immature neutrophils can be also seen under the microscope by proper staining. There are few, but they are very few in number. Mostly mature neutrophils, which are 60 to 70% of the white blood cells. 60 to 70% not among all cells, among the white blood cells are these mature neutrophils, which are size almost just greater than the size of the RBCs. Okay. So, whenever we identify the blood cells under the formed elements of blood under the microscope, what we find, those who have already seen in our hematology practical classes, that what you have seen, that so many RBCs and only few WBCs in the film, in the blood film which we are seeing under the microscope. So, how do we identify the cells? First we try to compare the cell size to the surrounding RBCs. So, what we see, WBC size, neutrophil size are just, neutrophil, eosinophil, basophil, these are just greater than the size of the RBCs. Again, basophil cannot be, basophils are very few in number. So, basophils are only, if we count 100 WBCs, maybe we do not find any basophil, or maybe find one. So, accepted value for basophil is 0 to 1% of the total number of WBCs, but all these are ideally will be called polymorphonuclear nucleosides because of the variation in the shape of the nucleus. They are writing here, they are writing 3 to 4 lobes, but still now we have, this is identified or this is accepted in standard textbooks as do not write 3 to 4 lobes, nucleus is 2 to 7 lobed. So, but some neutrophils, immature neutrophils, also called as band cells or stuffed cells, but they are not very easy to find. They are only say 2 to 3% maximum or 5% of the total number of leukocytes. Most neutrophils which you find under the microscope, we can observe under the microscope, are mature neutrophils. Whenever there is a very serious or very acute bacterial infection, then what happens? The body tries to recruit these immature neutrophils as well. In acute bacterial infections, if that time the blood is, say we collect venous blood and examine under the microscope, we collect blood and examine under the microscope, maybe at that time bad neutrophils will be visible and in more numbers, but usually bad neutrophils are just not even 5% of the total number of WBCs. And function of the neutrophils, as we told, mainly the phagocytosis, they release their granule contents along with these enzymes into the extracellular space, so that this process of phagocytosis and these invading agents can be taken care of, so that this chromatin material are decongested. They are in the extracellular space, they form a network where the bacteria are trapped and they are killed. So, phagocytosis plus killing of the bacteria by forming a network with the help of the decondensation of the chromatic material. So, the neutrophils themselves die during this process, but the body tries to recruit more and more number of neutrophils, even immature neutrophils at the time of, those are called band or stuffed cells during the time of acute infections. About the other granulocytes, eosinophil is its main. Eosinophil, again you have seen, it is almost the same size of neutrophils. Under the microscope we have told that this is actually like the nucleus is classically defined as spectacle shaped nucleus, bilobed spectacle shaped nucleus. They are just greater than the size of the RBCs, somewhat greater than neutrophils, but almost the same. Neutrophils, eosinophils, basophils, almost the same size. If we express it numerically, it is like 10-15 micrometer. They are writing 12 to 15 micrometer. Micrometer. This is micrometer. I mean 10 to the power minus 6 of a meter. So neutrophil. These are the things. This is the. It contains some proteins and some, again, neutrophils also contain, eosinophils also contain some proteins, and the enzymes. One major protein is in the eosinophil is called the major basic protein, and these granules, the neutrophils are granulocytes. The granules of the eosinophils are also granulocytes like neutrophils. The granules of the neutrophils are somewhat fine, but the granules of the eosinophils are somewhat coarse, and they take up, if we use Leishman stain, they take up the eosine part of the Leishman stain. Okay. So, those granules appear pinkish in color like the cytoplasm of the RBCs, and but most others pick up methylene blue. Methylene blue is another component of the Leishman stain. Most pick up methylene blue and are bluish violet in color, most components. But granules of eosinophils are coarse ones taking up the eosine stain, and they are, the colors of the granules are bright red or pink. Forget orange, bright red, pink. So, stains by acidic dye, eosine. So, this is the structure of the eosinophil. They are primarily involved in allergic reactions. Whatever, there are some other functions of eosinophils as well, but they move to inflamed areas in case of allergic reactions. These are the, you can call it the hallmark of allergic disorders. Okay, so they are primarily involved, eosinophils, in allergic reactions. They contain, that means they help the body to combat allergic manifestations. Okay. Again, basophils are very few in number, so difficult to identify under the microscope. Size almost the same of neutrophil and eosinophil, but as they are very, very few in number, and their granules, their coarse granules over, that is the overlying, the nucleus will be overlying the granules, and but most difficult to identify. Why? Because they are very few in number. Basophils contain some substances like heparin and all, which are there some role in phagocytosis along with the neutrophils, but they contain a substance called heparin, which is an anticoagulant in the body, and coagulation of blood. So, why is blood not coagulating in the body? So, heparin is responsible for that. They contain a factor called the eosinophil chemotactic factor, and thus basophil has also role in allergic reactions. They pull the eosinophils towards the area of where they should be the eosinophils content. This eosinophil content is required. That factor is called the ECFA, and the substance also secrete a substance called histamine, which is again a mediator of allergic reactions. One thing which they are adding right now is basophil itself. Heparin also like activates a hormone called the lipoprotein lipase, which is involved in lipid metabolism, mainly triglyceride metabolism. Lipoprotein lipase, you will read in biochemistry, facilitates absorption of triglycerides. So, like that, as basophils secrete heparin, so as heparin induces this lipoprotein lipase, heparin not induces, sorry, we should activates the hormone from the pro-hormone stage, activates the hormone lipoprotein lipase. So, indirectly they are saying that it has a role in lipid metabolism, but no direct role, because heparin has that role and heparin is secreted from basophil. So, they are writing it as a function of basophil, but mainly it is involved in allergic reactions like secreting histamine, secreting eosinophil, chemotactic factor, which like attracts the eosinophils to mediate the hypersensitivity reaction or the allergic reactions. Rest we are left with lymphocytes and monocytes. So, mainly you have seen in the practical classes, lymphocytes can be classified. Lymphocytes have a nucleus and there is a thin rim of cytoplasm around the nucleus. The nucleus is round shape, almost covering almost the whole of the cell, and the cytoplasm just forms a thin ring. There are some lymphocytes which are small and some lymphocytes which are large. Large lymphocytes are same structure, nucleus rounded with a thin rim of cytoplasm, but small lymphocytes are just greater than the RBCs, size just greater than the RBCs, but large lymphocytes are larger, almost greater than twice the size of the RBCs. From the immunity, that means point of view, large lymphocytes are also called something called the natural killer cells, and small lymphocytes are either T cells or B cells. What are T cells? T cells, B cell structure that cannot be identified by staining. Okay. So, they are not identified by staining. Small lymphocytes, T cells, B cells, we will read about them in immunity when we cover the section on immunity. So, small lymphocytes are those T cells, T lymphocytes and B lymphocytes. Large lymphocytes are the natural killer cells which are involved in innate immunity, and T lymphocytes and B lymphocytes in acquired immunity. What are those? We will have a discussion about that in the immunity section. And this is how the lymphocytes look like: NK cells, T lymphocytes, B lymphocytes, but these T and B lymphocytes can't be identified under the microscope by this staining method. Okay, all these helper T cells, cytotoxic types of lymphocytes, helper T cell, cytotoxic T cells, B cells, we will have a somewhat detailed discussion in the section of immunity. So, they are in the immunity portion. Monocytes are the largest WBCs. As you have attended the practical classes, by now you all know monocytes are the largest of the WBCs and having a bean shaped, kidney shaped, typically described as bean shaped or kidney shaped nucleus, which is towards one side of the cell and the rest of the cell is cytoplasm. So, monocytes contain abundant cytoplasm. These have no granules obviously, and it is important what is the functions of monocytes. This is one is after most important function is phagocytosis along with neutrophils, although which is the most important or the primary cell for phagocytosis obviously neutrophil, but monocytes are helping them. And what is it also acts as antigen presenting cell. Just know the term for now, antigen presenting cell will be discussing along with immunity. And monocytes become macrophages. They remain in the peripheral circulation maybe maximum for a few days, and then they become macrophages and enter the tissues. Monocytes are these macrophages are transformed monocytes which go from the peripheral circulation into those various tissues which are associated with our immunity. Monocytes, they secrete various chemical factors, they are known as chemokines, but main function, that means they are the second line of action for phagocytosis after neutrophils. Okay, this is again in just a summary of these structures of the different types of WBCs, which you have already, most of you have noted on the microscope. For those who have joined newly, we will try to arrange a few classes for you, 1 or 2 classes, so that you can get acquainted with those things. Okay. So, this is more or less about the WBCs. They are adding a section called regulation of the WBCs. So, it is not very clear to me as well what they are trying to say by regulation of WBCs. Regulation of, it may be regulation of, like regulation of erythropoiesis are by this hypoxia and the erythropoietin and these dietary factors as we talked of. Regulation of, if they try to mean regulation of granulopoiesis, then whenever these WBCs are decreasing number, most things in the body, most things in the human body for the purpose of homeostasis. So, we had a class on those who are joining newly don't know about it, but you have to read about, there is a section of homeostasis in general physiology, mostly are occurring through negative feedback mechanism. So, whenever granulocytes are decreasing number, there is some granulocyte releasing factor, some granulopoietin, which stimulates the bone marrow to cause the increase in the number of granulocytes, and then when this happens, again some factor which has not been identified, mature granulocytes release that factor, so that bone marrow, again this stimulation is, this bone marrow again not suppressed, but this overstimulation of bone marrow is prevented, and the number of neutrophils are maintained, or the number of granulocytes are maintained where it should be. Okay, that means for which is optimum, which is not too much, which is not too less. So, one thing which we have no much, we don't have much more time, but what we like to discuss is next week we will be having practical sessions on blood grouping. So, blood groups, as you all know, blood groups were this ABO system of blood grouping. What is the preliminary idea? You should have a knowledge of it. So, blood groups are these are the 4 types of blood groups according to the ABO system. Just what is the preliminary idea? That is, blood groups, these groupings are done on the basis of the presence of antigen and antibody, better known as agglutinogens and agglutinins. Okay, agglutinins are present in the blood plasma, agglutinogens are present on the surface of the RBCs, and this naming is according to the presence of agglutinogens. Like A agglutinogen is present on the RBC of a person with blood group A. B agglutinogen is present on the RBCs of a person with blood group B. O blood group means none of the agglutinogens will be present, and AB blood group means both A and B agglutinogens will be present. Now, the person who classified these blood groups was a person called Karl Landsteiner, and he did it in 1900. Okay. So, what he told was that, and what he told means what is a person containing, those are called the Landsteiner's laws, that whatever blood group is, whatever antigen is present on the surface of the RBCs, the corresponding agglutinin cannot be present in the blood of that person. So, that person's plasma will not contain the corresponding agglutinin, which is alpha. So, what it will contain? Plasma will contain beta agglutinin. For B, beta cannot be present in that person's plasma. So, is present is alpha. Wherever no agglutinin is present, both agglutinins are present in case of blood group O. And for AB, as A and B agglutinogens both are present, it cannot have any of the agglutinins in blood. So, this is the preliminary idea based on which your practical sessions on blood groups, you have to identify blood groups. So, what happens is that whenever in case of you will get artificial substances which contains these anti-agglutinogens, those who will be This they are called the antisera, anti-agglutinogens, which will agglutinate these agglutinogens. Okay, so you will get these colored substances called these antisera, which will contain, say, a substance which contains this alpha will agglutinate A. So whenever this agglutination, which can be seen with naked eyes and better under the microscope, so this can be seen with naked eyes, if this agglutinate the blood, then we can conclude which blood group it is. If it is agglutination with both A and B, then it is 8B blood group. With none, this is O blood group. And there is another antigen or agglutinogen identified, that is the D, that is the rhesus factor, to which no naturally occurring agglutinins are there, but there is this artificial antisera. When you react with them, this will confirm whether the blood group is Rh positive or Rh negative. In the next week, we will do it in the practical session.